Today we're going to be learning the basics of neurons. Before we start studying neurons, let's learn the basics of what a neuron is and what they do. The definition of a neuron is an electrically excitable cell that communicates with other cells through areas called synapses. Neurons are the main component of nervous tissue. They're found anywhere and in between all types of nervous tissue. Now that you know all the definitions and what a neuron is, let's get into this basic structure of a neuron. The structure of a neuron is fit for its function. The first part of the neuron is dendrites. Dendrites are the web-type structures in the front and on the sides of a neuron. The second part of this neuron is the cell body, which is an oval-like structure in the middle of this neuron. Then is an axon where the electrical signals travel through to the next neuron. The membrane that is wrapped around the pathway is called myelin sheath. It is composed of lipids and proteins, which is a plasma membrane. The myelin sheath is wrapped around the axon till the end of the neuron, or to the end of the axon. Now let's take a closer look at the axon. This membrane is polarized with positive potassium ions on the outside and negative sodium ions on the inside, and it stores energy of the signal like a battery. Polarized means there are slight charges, either negative or positive. There are many starting points for a signal to be passed on a neuron. Although there are many starting points, there is only one exit point, through the one and only axon. Dendrites are the initial place where a signal will be received on a neuron, which will then be passed down through the axon to the next neuron. The axons are coated with Schwann cells, which inside them are layers of myelin, and at the center or core is the axon. The Schwann cells are connected by nodes of Ranvier. In this picture, it shows there's two Schwann cells, and in between the two is a yellow or yellowish orange segment, which is called a node of Ranvier. Within the Schwann cell, there's a nucleus of the Schwann cell, and within that, there's layers of myelin, and at the core is the axon. The main functions include protecting the nerves from other electrical impulses and shortens the time it takes for a nerve to travel through an axon. Nerves without the myelin sheath must send a signal down the entire length of the nerve. In large organisms, this presents a problem as the signal can take a long time to reach the end of the nerve. Due to salutary conduction, salt concentration, the individual myelin sheaths cause a signal to leap from one node, which is a segment of Ranvier, to the next. The myelin sheath does not allow the action potential to form where it covers the axon. Instead, the electrical wave created at each node speeds at the action potential to the end of the nerve where it can be transferred to the next neuron. This mode of transmitting a signal down the axon also saves energy, as the sodium and potassium pumps that regulate the action potential are not needed under the myelin sheath. The sodium ions and potassium ions switch places, which causes the neuron to send the signal down through the axon. Now let's talk about how a neuron sends the signal to the next neuron. The process of a neuron transferring its signal to the next neuron is called synapse. The synapse is the site where a neuron's dendrite and a neuron's axon interact. So first, the action channel or action potential depolarizes the membrane, which means it depolarizes means it loses the charge of the membrane. Next, after the depolarization of the membrane, the calcium channels open up to release the neurotransmitters, which are in the synaptic vesicles, which then are brought outside of the axon terminal into the place called synapse, which is between the axon of the present neuron and the next neuron's dendrite. The neurotransmitters are then passed through the myelin through the diffusion. Diffusion means pass through a membrane without the help of any proteins or any other internal or external force. Then on the receptor neuron, The ion-gated channel proteins, which are the receptor proteins on this diagram, open up to let the neurotransmitters in. Then once the neurotransmitters are passed through, it is either broken down or reabsorbed into the neuron. Neurons with the myelin sheath send signals at high speeds, while neurons without the myelin sheath send signals at slower rates than the ones with myelin sheath. As shown in this diagram, the one on the left is what a neuron would look like if it did not have layers and segments of myelin sheaths. And the neuron on the right is what a neuron would look like with layers and segments of myelin sheath. As you can see on the neuron example on the left, the electrical current or the impulse is very much slower than the one on the right. It is because it is lacking the myelin sheaths or Schwann cells, and it is also lacking the node of Ranviers. The node of Ranviers and the Schwann cells allow the impulse to jump from cell to cell at a quicker rate than just letting the impulse or electrical charge travel down the axon at a normal rate without Schwann cells and the segments called node of Ranviers. The neuron and its electrical message is an example of a signal transduction pathway. A signal transduction pathway is when a message is sent from a cell to another cell, which ultimately leads to a cellular response. This chemical message attaches to the ligand then, which releases an enzyme that would cause a protein to work that would then release a secondary enzyme. The second enzyme would then open the ion channel, which would release sodium ions in the neuron. In addition, there are numbers of things that can affect a neuron's ability to work. For example, drugs like marijuana and alcohol can affect a person's response time. These drugs have things in them that interfere with the ability of the neuron to work properly. Finally, there are other drugs like metamorphines or other venoms in snakes and spiders that can block neurons from working, that can block feeling pain, and can even cause the body and even the brain to shut down. This diagram shows how a signal transduction pathway takes place and all the process of how it occurs. Growth factors and other messenger molecules that bind with the receptors on the outside of the cells activate genes deep inside the cell. This happens because the messenger molecule activates a chain of events that carries a signal through the cell to the nucleus. When a growth factor binds it to its receptor on the outside of the cell, which is the red, which then binds to the purple, which is the receptor cell, the receptor transmits the signal to the inside of the cell. That step is called reception. That signal activates the first molecule, which then activates the next one, which that activates the next one, and which ends in an activation of a protein called transcription factor. And that step is called transduction. The last and most important step is the transcription factor then binds to its target gene in the nucleus. It activates the gene and initiates the process of protein synthesis. Signal transduction pathways tell healthy cells to divide at the right time. However, gene mutations can lead to abnormal growth signals, causing cells to divide when they should not. This leads to cancer. Now that you know the functions and importance of a neuron, let's get to the fun segment of the video. The questions! So take out a piece of paper and write down the answers to the following question. Question 1 is, what is the structure of a neuron? Question 2 is, what is the casing that is wrapped around the axon called? And question 3 is the transfer of signals through the neuron efficient or not? Explain why.